So let's solidify some terms. So haploid versus diploid was something I just kind of referenced but didn't really explain. Most of the cells in your body are diploid. I kept saying we're diploid, but really most of the cells in your body are diploid. And diploid again means you have both sets of chromosomes. Okay. Haploid, on the other hand, means you have half of your chromosomes, or half of the chromosomes of a diploid individual. So when do you see haploid cells? Most of the time you see them in sperm and eggs. We're going to talk about this later. You can also see full-on haploid organisms in things like ants and things like bees. But we're gonna worry about this later on. What I want you to know is that most of your cells are diploid. Some of your cells, like your sperm and egg cells, are haploid, meaning they have half of the chromosomes that they need. We also have this notion of homologous chromosomes, which are matched pairs of chromosomes, which hopefully you remember from the last lecture, means you got one from your mom and you got one from your dad. The reason I spent all that time yammering on about DNA and stuff is because the topic of these lectures is mitosis. Mitosis is just making a copy of a cell, and in order to make a copy of a cell, you need instructions, right? You have to have instructions for what's going to make those proteins that are going to allow each of those cells to live. In order to do that, we're going to have to copy our DNA, which is actually going to be a topic that we cover later on, and we're going to talk about how the mechanism of copying DNA works, but we're not concerned with it right now. What we're worried about is mitosis, making a copy of a cell. And you might ask yourself, or me, why? Why would we need to make a copy of a cell? What difference does it make? Well, think about the last time you scraped your knee on the ground. Where did the new cells come from? Where did those new cells come from to cover up that wound that you created with the concrete? They came from mitosis, okay? So whenever you need to make new cells, mitosis is there. Or think about the size that you were when you were an infant. Why are you not infant-sized anymore? Well, because you grew thanks to our old pal, mitosis. I would also like to clarify right now, because this is always a problem, we are not talking about meiosis. And this is another case of, well, this looks a lot like this one, but these two things are different. We're going to talk about meiosis later on, but right now, all we're talking about is making a copy of a cell. So this word meiosis can be in your head, but it is not what we're doing. Okay? We are making copies of cells, that is all. Mitosis is just one part of what the cell is up to in its overall life. And what the cell is up to in its overall life is generally referred to as the cell cycle. And mitosis is going to be part of that, but hold on. First, we need to talk about how is it that the cell gets ready to duplicate itself. How does the cell get ready to make a copy so that you can grow, or that your wound can be healed, or that your old cells can be replenished? Well, the answer to that is interphase. Interphase is where your cell is spending most of its time, by the way. And interphase is broken down into several phases itself. We have the G1 phase. By the way, this is called gap 1, not growth 1. The book kind of doesn't do a great job of differentiating that, but this is called the gap 1 phase. And in gap 1, basically your cell is getting ready to replicate. It is building up enough of those nucleotides and phosphates so that it can replicate its DNA, which we're going to talk about in just a moment here. And it is getting ready to duplicate DNA so that it can split. Remember, you need to have 2 copies of DNA in order to have 2 different cells. The next phase in interphase is the S phase. S stands for synthesis. S stands for synthesis. But what are we synthesizing, Aaron? You are synthesizing DNA. You're making a copy of your DNA. Okay, so in S phase, you're making a copy so that you can have a copy for the new cell to show up. Again, we are going to talk about the process of DNA replication in detail later on. For now, it is sufficient to know that you are making copies of your DNA. So let's draw another one of those homologous chromosomes here. So we have one here. This is again, I guess my mom's or my dad's. I don't remember the color scheme I used. It doesn't matter. But here's one copy of, let's say, chromosome 15. Here's the copy I got from my mom of chromosome 15. These two things are called homologous chromosomes. Okay, remember that. These are called homologous chromosomes. They have similar genes in similar places. But during S phase, we're going to make a copy of these things. And that's getting to our sort of more generic looking chromosome things, those X's that you're going to see in 23andMe. I'm going to draw these better. I fixed our chromosomes up here. So remember, this blue one is the homologous chromosome of this pink one. But now we've made copies. Okay, and each of these copies is referred to as a sister chromatid. sisterchromatid. Sister chromatids. Sister chromatids are exact copies of one another. Okay, this is an important distinction. It's going to be on a quiz or a test or both. So sister chromatids are exact copies of one another. I'm going to show you in a few minutes why that is, maybe in the next lecture. Sister chromatids are exact copies of one another. Homologous chromosomes are different chromosomes that have similar genes in similar places. Remember, I got my mom's tall gene on a chromosome, and then I got my dad's short gene on its homologous chromosome. Okay, so that's important to remember. Sister chromatids are not the same as homologous chromosomes. Again, interphase can be broken into these sort of 3 steps. So you have G1, which is basically can be thought of as getting ready to replicate your DNA. So we'll say getting ready to replicate. Okay, it's gathering up nucleotides, maybe it's building up some energy. It's just doing things that allow it to get ready to copy its DNA. And again, that's gap 1. S is synthesis. And in synthesis, we are copying basically the cell is copying, I'm saying R, but it's copying its DNA. So in S phase, the cell is going to copy DNA. Why are we copying DNA? Because we're trying to replicate the cell itself. And so we need 2 sets. Almost forgot, we're also copying something called the centrosome. We're going to discuss the centrosome later, but the centrosome is basically going to help the DNA to split. It's going to help the cell to actually split apart. Then we have the G2 phase, G2, which stands for gap 2, and basically this thing is getting ready to split. The cell is getting ready to split. So it's starting to make proteins that the cell is going to need in order to split in half. It's replenishing energy that it used up during the process of DNA and centrosome synthesis. And this sort of takes us to the end of interphase. But you can think of the interphase as just doing its normal cellular processes as well as getting ready for mitosis.